Good morning, everybody. Thank you very much, Jennifer, for inviting me here. It's a privilege to be here. My talk will be slightly different. I would like to share with you some thoughts about diseases of poultry. Our laboratories at the vet school, Dr. Van Santens and I, we work on trying to produce knowledge to protect poultry so that it is still, well, and it remains, the most important protein source for human consumption worldwide. Indeed, we eat a lot of chicken and eggs in the world. There are several diseases affecting the poultry industry, and as I said before, we try to create knowledge to prevent these diseases. One of the most important diseases, according to this report, is infectious bronchitis virus. As you see here, 60% this thing doesn't work. But the one that says IBV, it's infectious bronchitis virus. This is the economically most important disease, and the agent is shown right there. These virus particles, which look like a crown, those are coronaviruses. Those are responsible for that disease. And today I would like to explain you why is this virus so successful. The immune system works similar as the police system in a country. If the police has a good picture of the bandit, they will catch him very quickly. If they don't have a good picture, it's going to be more difficult. The immune system is exactly the same thing. When we vaccinate, we provide the immune system with a picture of the virus, and it will be destroyed. How does this virus escape immune responses induced by vaccination? This is what I wanted to talk to you today about. So it's interesting how the immune system is really the same as the police system, because, you know, we have a border control that's innate immune responses where they just check the visas if you have a passport. But then you have criminal police. They will go for the terrorists. If they have a picture of them, they will immediately catch them. So there are several strategies by viruses to escape the immune responses. And this is probably the most fascinating one. There are other strategies, for example, to be extremely resistant, that you can't destroy the virus. Or to be transmitted from the mother to the progeny, which happens, of course, in many animal species. But changing the look of the virus is probably the most amazing. And this is what avian influenza viruses do and coronaviruses do. Just to understand how this works, I will try to explain something very basic that most of you know. So proteins are the most important structural units of animals and everything, plants and bacteria and viruses. The smaller units of the proteins are called amino acids. And the sequence of those amino acids, which will determine what type of a protein we have, depends on the DNA. And so if you change the sequence of the DNA, you change the amino acids and you change the protein. Again, so these viruses, what they do is change their face. They change that feature of them that will be recognized by the immune system. You know, if you change your face, if you're an outlaw, you will not be found by the police. You know, if the police gets a portion of you that is very conserved among humans, like a picture of your liver, that won't help them. It helps to find, you know, the face. That is what actually varies. And as I was saying before, if you change the amino acid sequence, this doesn't work either, then you change the protein, then you have different structures. And I'm just showing you these different phenotypes, which are called these different structures of the same species, in this case, peach trees. Some of you will recognize some of these as peach trees, some of you may not. In this virus, we have the same, but we can't see those flowers. We just can't. So we can't differentiate between these two different strains that you are seeing here. IBVQX and IBV-ARC. They look the same for us. So because we can't see those differences, we do the sequencing of some proteins of this virus. And for example, here it shows you, this is how we do it, the sequence chromatogram. We see that the first amino acid is leucine, and the second is tyrosine, the third is glycine. We see then that they will have a particular shape. And this is because of these different nucleotides having, those peaks in the sequence chromatogram. In this coronaviruses, what changes is the spike, which is these corona-like structures on top, on the envelope of the virus. Those are the ones that really change. So if we were able to look very closely, we would be able to see differences between these viruses. And indeed, when this is a model that shows that by changing these few amino acids shown below in the chart, in the table, changes actually the structure or the phenotype of the spike. So the police, the immune system, will not be able to recognize by just changing a few amino acids. And the mechanism behind this was discovered a long time ago when Charles Darwin proposed the principle that the preservation of favorable variations and the rejection of injurious variations he called natural selection. That's how these viruses work, and every species, by the way. Trying to show you how it works for this virus. So evolution by natural selection can be divided into two steps. One is the generation of genetic diversity, which is independent of selection, and the second is the selection process. The generation of genetic diversity in bronchitis virus is by two mechanisms. One is mutations, and the second is recombination of the genetic material. So we have a genetically diverse population, as shown in that picture. The first mechanism, mutations, happens when the enzyme is putting nucleotides one after the next, so that distinct amino acids will be encoded. And the protein will look like a particular thing. But sometimes it looks like this machine right here, which is putting bricks in line. Sometimes that machine will not find a brick, and we will have a deletion. The protein will look then different. If the machine picks a block instead of a brick, we'll have a substitution. And if it picks a brick that has a different color, it will also be a substitution. So that changes the genetics of the population by mutations. The second mechanism is when two different strains infect the same host cell. Then the progeny of these viruses will be, one will be exactly like one parent, like A. The second will be the same as the other parent, like B, but there will be some viruses sharing both features of both parents. Just like in human populations. Because recombination is sort of a way similar to sexual reproduction. There will be exchange of genes. And by the way, genetically diverse populations are the ones that are stronger. Those are the ones that will be able to adapt changes in the environment. Is this true for this virus? Yes, indeed it is. This is 3 different viruses that originate from the same original viruses. Actually, these are 3 vaccines. They all come from exactly the same virus. But as you see here in vaccine A, there is 2 peaks right there. So you have a major population encoding tyrosine and a minor coding histidine. Vaccine B has only tyrosine there. And vaccine C has a major population coding for histidine and a minor coding for tyrosine, even though they come from exactly the same original virus. So we have a genetically diverse population, just like other populations. That was step 1. The second step of natural selection is the actual selection process, where the genetic endowment of the few survivors during reproduction and thus abundant new genotypes are tested in the next generation. Individuals favored by selection will contribute genotypes to the gene pool that were apt to spread in future generations and thus enhance the adaptation of the population as a whole. So we start with a genetically diverse population like that one. We apply selective pressure, and we may have this result, for example, that particular individual was more fit in the environment, or we may have this population, which is a mixed population, where two individuals were more fit in the environment. That's the same with other species, with all kinds of species. For example, the white skin color, for example, is an adaptation to low levels of sunlight, because white skin will allow the sunlight to go through, and you will be able to produce vitamin D. So people living in areas where there was dark areas in the world, Norway, Finland, Northern Europe, you would be better adapted if you have white skin. In contrast, black skin will block the sunlight. If you live in areas where there's lots of sunlight, you want to have black skin, because then there will be enough vitamin D production in your skin cells. If you put a black person in a low sunlight area, you will have more a risk of a deficiency of vitamin D. If you put a white person in lots of sunlight, you will have skin cancer. Is this true for viruses? Here we put a virus into these chickens by eye drop. And you see there is, the sequence of that virus shows two peaks in the middle. One codes for serine and the other for alanine. When we rescue this virus out of the chickens afterwards, we find that just one population was more fit, was more successful in that environment. It codes only for alanine, so selection works. This is why we have all these outbreaks of disease everywhere in the world with different strains of bronchitis virus. And the greatest challenge for IBV vaccines is to protect against a continuously changing target. Thank you very much for your attention.